Hey guys, welcome back to One Stop Biology. I hope you guys are doing good. So, guys, today we are going to start off with the topic of AIDS. So, let me remind you that we are right now at the chapter 8th of class 12th NCERT, which is Human Health and Diseases. And we have already covered many, infectious diseases. We have covered immunity. We have covered different types of immunity that our body acquires. Then we covered the immune system in the last video. So today we are going to study about another, chronic disease, which is AIDS. So guys, AIDS stand for acquired immunodeficiency syndrome, right? So this basically means, what does it mean? This means deficiency of immune system, right? I- The word is acquired immunodeficiency, right? So the immunity that we acquire is deficient, basically, right? So it basically means deficiency of immune system. Now, this, what kind of immune system? The immune system which is acquired during the lifetime of an individual. So, the immune system which is acquired during the lifetime of an individual, right? So basically, what does it mean? What does it indicate? This basically indicates that it is not a congenital disease. It is not a disease which is there since you are born. Congenital diseases are such diseases which acquire when you're born itself. You have these diseases as soon as you are born. You're born with these diseases. AIDS is not a congenital disease, right? And what does the term syndrome? What does the term syndrome means? Syndrome means group of symptoms. Syndrome means a group of symptoms. So let's first see what is the history behind AIDS, guys. So AIDS was first reported in 1981. AIDS was first reported in 1981, and then it has spread a lot all over the world, killing more than 25 million people. So 25 million people, this is the reported data, guys. So people have been killed because of AIDS since 1981. So AIDS basically is caused by the virus. The virus is human immunodeficiency virus. It is caused by HIV. Full form of HIV being human immunodeficiency virus, right? So this basically is a group of retroviruses. AIDS is caused by HIV, human immunodeficiency virus. And these are retroviruses, right? So the virus has an envelope which encloses an RNA genome. So basically, it is a type of virus which has an envelope, and inside that envelope, you have the RNA of the virus, right? So here, the structure that you see, this, and inside you have the RNA. This is the retrovirus. Right. Now, how does, HIV gets transmitted? How does it goes from person to person? So it occurs by various factors, guys. Right. It even occurs by sexual contact. If you basically come in contact with a person, infected person, if you come in sexual contact with a infected person. It means if you copulate with an infected person, you get HIV. You can get HIV. If the infected person's basically blood contains the virus. So if that infected person goes and donates blood to the blood bank, and that blood is transfused in a healthy individual, it might cause HIV. If you go to a hospital for an injection and that injection has been used before, maybe the injection has been given to an infected person, to a HIV infected person, and if that same infection is used giving injections to you, you might have HIV. And this happens a lot with the drug abusers. So, a lot of drug abusers take the drug through these injections. So they inject it through IV. Basically, they take it direct to their veins and they share these needles. So again, HIV and AIDS is very common among drug abusers. Also, it can happen from an infected mother to the child through placenta. So there are a lot of reasons, there are a lot of means through which HIV can be transmitted. One being sexual contact with an infected person. Two, if blood transfusion of HIV contaminated blood, if this happens. Third is sharing infected needles. And fourth is from infected mother to her child through placenta. So these are the means, guys, through which HIV can be transmitted from an infected person to a healthy person. Now let's see what is the process, how the retrovirus exactly, replicates. Let's see this process as well. So as you can see in the diagram, let me show you through the diagram itself. So if you see the diagram here, what is happening exactly? So first what is happening is that you have a retrovirus here. You have a retrovirus here, right? This is the RNA. So the outside is the protein coat and inside you have the viral RNA. Now this comes and this infects a normal cell. So say this here what you see is a normal animal cell. The outside you have plasma membrane, right? So this retrovirus comes in contact with the animal cell and the viral DNA, the viral RNA is introduced into the cell. The viral RNA from the protein coat, from the viral protein coat comes out and it just enters into the animal cell, right? Now after it enters into the cell, what happens is that by reverse transcription the RNA is converted into DNA. That is why, guys, these viruses are known as retroviruses. Why? Because they can produce our DNA from RNA by reverse transcription. So by reverse transcription, once they enter into the animal cell, they produce viral DNA, right? This viral DNA that then comes and gets incorporated into the human genome, in the host genome. It goes inside the nucleus basically, and it gets incorporated into the healthy DNA of the person, of that person. And then this starts producing a new viral RNA and this cell becomes infected. So this cell becomes infected and now it starts producing a new viral RNA. And these viruses are now basically produced and they starts multiplying, right? So basically your nucleus starts producing new viral RNA and it starts multiplying. And again, it goes outside the body and through sexual contact, through blood transfusion from an infected mother to her child, this virus can infect other cells as well. Now, remember one thing, that infected cells can survive, right? While viruses are being replicated and released. So, here you can see a note. Now, what does it mean? That once the new viruses are formed, this doesn't mean that this animal cell, this whole cell, is going to disintegrate or something like that. This cell is going to survive, right? So basically, these RNAs are not killing these cells where they are replicating and they are multiplying. They are not doing anything to that animal cell, right? They are easily replicating and they are released outside while the animal cell is surviving, right? So this is the entire process of replication of retroviruses, right? This is how they reverse transcriptase, they reverse transcribe, they form DNA. That DNA gets incorporated into the human DNA, that DNA of that particular person, and then it starts producing new viral RNA, which gets multiplied, it replicates, it multiplies, and it goes and infects other cells as well, right? So guys, this is all about the replication of retroviruses, right? Now we have already seen how they are transmitted. So they are transmitted by sexual contact, transfusion of contaminated blood, sharing infected needles from infected mother to her child, right? So guys, always remember that there is a time lag between the infection and the symptom of that particular infection, right? Now in the case of AIDS, in case of symptoms of AIDS, this period may vary from a few months to even 10 years, right? So in many people what happens is that they get AIDS and the symptoms starts coming after 10 years, 10 long years, right? So what happens basically is that after getting into the body of that person, the virus enters into the macrophages, and these macrophages, what happens is that the RNA genome of virus replicates to form viral DNA, and then with the help of this happens with the help of reverse transcriptase, right? Now that gets incorporated into host cells, and that produces more virus particles which starts infecting more and more macrophages, right? So basically the macrophages are considered as the HIV factory, right? And then what happens is simultaneously they enter into the helper T lymphocytes as well, right? So T lymphocytes, we studied about P lymphocytes and T lymphocytes, right? They basically provide us acquired immunity. Am I wrong? No, right? So basically what happens is simultaneously they are getting produced in macrophages, and then they start getting into T lymphocytes as well, and they start replicating there as well, and they start producing progeny viruses there as well. Now these progeny viruses are released in the blood and they start attacking other T lymphocytes, right? Now this is basically repeated, leading to a progressive decrease in number of helper T lymphocytes in the body. And as soon as it starts, decreasing the number of T lymphocytes, basically what they're doing is they are weakening your acquired immunity, because T lymphocytes helps the B lymphocytes in producing antibodies, right? So basically they are the ones which are giving you acquired immunity against any particular microorganism or pathogen. Now, as they start attacking other T lymphocytes, the production of T lymphocytes is decreasing, and the basically disease is progressing now in your body. And as it starts progressing, as soon as it starts even infecting more and more cells, your body's immune system is weakening, right? And then this is the time when you start showing various symptoms, like you have fever, you start getting fever, you have diarrhea, you have weight loss, right? And then what happens, and since your immune system is now very weak, you any kind of infection that you get, either from bacteria, viruses, fungi, or any parasite as such, your body basically starts suffering from these infections because your immune system is compromised and your body is struggling to fight from any kind of infection now. So any kind of infection, if it happens in your body is basically not able to fight these infections now. And at the end, the patient becomes so immune deficient that they are unable to protect themselves against any kind of infection. So you are not at all able to protect yourself against infections, right? And that is when the immune system of a person stops responding completely and it leads to death, right? So there are basically, guys, a lot of diagnostic tests. There are various different types of diagnostic tests for AIDS. One of the tests is known as ELISA. ELISA means enzyme linked. Enzyme linked immunosorbent assay. ELISA means enzyme-linked immunosorbent assay. So this is a diagnostic done, which is done this diagnostic test, which is done on any person which is, highly likely to have AIDS because of his or her symptoms. Now, there are several treatments as well. There are several treatments as well against AIDS. So there are antiretroviral drugs. There are certain antiretroviral drugs which, but what happens is that it doesn't cure your disease. What it happens is that it is partially effective and it can prolong your life if you have AIDS. Right. But remember that basically what it is giving you is it is giving you some more years to live, but it does not prevent, it cannot prevent death. Right. So there are antiretroviral drugs which are used on as treatment for the AIDS infected individuals, but it can only prolong your life. It cannot cure the disease and it cannot prevent death. Okay, and that is why the saying comes that prevention is better than cure. So in case of AIDS, that is why it is said to be aware of what are the what are the causes, and you have to prevent yourself from coming into those situations where you can have AIDS. So one of the major cause of AIDS is having multiple sexual partners, right? And if you're not taking proper precautions while having sex, while copulating, so one of the symptom is basically one of the cause is having multiple sexual partners. So that is why it is said that one should avoid having multiple sexual partners. Now again, the other cause was through infected needles, right? So within drug addicts who take drugs intravenously, it happens a lot of time that they get AIDS. So again, needles, avoiding used needles at any cost. Even if you are visiting a hospital and you are about to get an injection, you should ensure that the needle is a fresh one and is not used before, right? Again, the other way is through contaminated blood transfusion. So basically, whenever someone is donating blood or getting, blood transfused in their system, they should make sure that if you're going for a blood donation, you should make sure that you go for an HIV test first, and once the person is negative, then only they should donate blood. In the other case, where you need blood transfusion, then again one should make sure that the blood that is being transfused is tested properly against HIV, and you should check by yourself that it is mentioned there that it is checked for HIV infections, right? So all these precautions are very, very, very important. And if you imply it in your daily life, it is not that difficult as well. It's just that one must be aware of the chronic, problems that are going to happen if you're not aware of these things, if you're not preventing yourself from these problems, right? So, guys, I hope you understood the basics of the disease AIDS. You understood what are the symptoms. You understood what is the cycle of the virus, what kind of virus, and how it infects the cell, the host cell, and what are the prevention of AIDS. So, guys, please do like the video, share it with your friends, and yes, if you are new to the channel, please do subscribe to the channel. And guys, if you have any doubt in regards to this video, please do not hesitating in asking me your doubts. You can either mention it in the comments or you can also message me on WhatsApp and ask your doubts. WhatsApp number is given in the description of the video. Okay, guys, see you in the next video. Thank you for watching this video. Bye bye.